Yes, very happy to continue the session to present our breast tumor-on-chip model that we apply to assess the efficacy and safety of CAR T-cell therapy. My name is Ibrahim Maulana. I'm a PhD student at the University of Tübingen, Germany, in Peter Losk's lab. So, CAR T-cell therapy is a type of cancer immunotherapy that uses genetically engineered lymphocytes to express chimeric antigen receptors, or CARs, to target specific antigens on the tumor. So the therapy has been quite a success story for various types of blood cancers, but not for solid tumors yet, and this is due to several challenges. The first challenge is related to the efficacy, because in solid tumor, the CAR T cells really need to operate within a dense and immunosuppressive microenvironment, which exhausts them over time. And in terms of safety, the therapy has been associated with adverse effects. One of the most prominent one is called cytokine release syndrome, or CRS, a condition where a high inflammatory cytokines are being released systemically in a certain subset of patients, causing organ damage and eventually patient's death even. So it is important to note that there is so far no existing model to basically predict the occurrence of CRS in patients and to study the mechanistics really behind that. So these are kind of the aspects that we would like to recapitulate within the tumor-on-chip model. And today I would like to give an example of how we can use the system to model acute CRS management by modulating CAR T-cell activity during the therapy, as well as testing whether we can capture patient-specific effects by integrating patient-derived breast cancer organoids. So for that, we designed this tumor-on-chip model. It is a chip composed of two stacked microchannels separated by a porous membrane. The bottom channel consists of 6 individual cylindrical chambers, which we then load with either tumor aggregates from cell lines expressing the target antigen or patient-derived organoids embedded in a synthetic gel. And above these individual chambers lies the media channel, where we then mimic the endothelium by adding endothelial cells either from primary derived or iPS derived. And then through this media channel, then we perfuse the CAR T cells or control T cells from the same donor, where they're then able to extravasate and then migrate down to the tumor site. So to go more into detail into the experimental design that we typically do, we use CAR T cells that are specifically targeting ROR1 antigens. So we expand the target cells, for example, the MDA-MB231, because of their overexpression of ROR1, tagged with GFP, or the patient-derived organoids, and then expand the endothelial cells as well. And then we load the cells into the chip and establish the tumor-on-chip model, and at the same time expand and thaw the CAR T cells. And day 0 mark the starting point of the therapy. Here we perfuse either the ROR1 CAR T cells or the control T cells, again from the same donor. And we do the administration CD4 and CD8 in 1:1 ratio, so mimicking also clinical administration here. And after that, we continue the culture for up to 8 days, where we then sample the effluents for measuring the cytokines, as well as the monitoring the interaction between the T cells and the tumor. So what we then saw is, for example, within the first 2.5 hours, we could see that the CAR T cells actually migrated towards the tumor aggregate faster compared to the control T cells. As you can see that the CAR T cells are going towards the focal plane of the tumor aggregates faster, whereas the T cells, most of them are still located in the media channel during this period. But then when we prolong the culture, we could see on day 1 that the control T cells were actually able to penetrate into the tumor aggregate. But in the CAR T cell condition, we could see a significantly higher density of infiltration, as you could see here. Here we measure, for example, the intensity, mean intensity of the T cells within the tumor region of interest, and it is also significantly higher compared to the CAR T control condition, where we perfuse the CAR T cells through chips containing ROR1 negative fibroblasts to assess also off-target effects. And this is consistently happening throughout the 8 days. So we had high density of infiltration of CAR T cells on day 1, 4, 6, and 8, basically. And what we then saw on day 8 is that the tumor aggregate actually continued to grow in the control T cells condition, whereas the growth of the tumor aggregate in the CAR T cell condition was hampered. And as the tumor aggregate are expressing GFP, we could basically also quantify the signal before and comparing before and after treatment to basically measure the lytic activity done by the T cells. And here we could see that in the CAR T cell condition, the GFP signal dropped after 24 hours, whereas in the control T cell condition, it remained relatively the same and even increased. And during all these interactions over 8 days, a lot of cytokines are being produced. So because we are interested in modeling cytokine release syndrome, we measured the typical cytokines, also measured clinically to basically tell whether or not the patient will develop cytokine release syndrome or not. For instance, IL-2, IL-6, IL-10, TNF-alpha, interferon gamma, and granzyme B. What we could see here is that the cytokine level consistently peaked on day 1, so after 24 hours of the CAR T cell administration, and then went down over time in the CAR T cell condition, which is in red. Whereas in the control T cell condition, the cytokine level remained stable and below the level of the CAR T cell condition at almost all measurement time points. So what is interesting here is that we were actually able to mimic the not only the level of the cytokines being released clinically, but also the kinetics of the cytokine release happening in patients that suffer from cytokine release syndrome. So moderate cytokine release is actually good and predict efficacious clinical outcome, but too much and too long could be deadly for the patient. So in the clinic, there are several ways how to basically manage that. For example, using IL-6 inhibitor or IL-1 inhibitor. But what we would like to test here is to actually interfere directly with the CAR T cell activation signaling to basically prevent them from secreting the inflammatory cytokine release and basically causing subsequent activation of, for example, macrophages and the monocytes and cause, in the long run, cytokine release syndrome. So we would like to check whether we could use this to manage the cytokine or the inflammatory cytokine response, right? So first we tested whether or not this will impact the viability of the CAR T cells, which is not the case. And then we also checked whether or not it could really deactivate the CAR T cells. Here, for example, shown the expression of CD69, which is the early activation marker of the CAR T cells. We could see that the MDA MB231, which is the target cells, actually upregulate this expression, and we can basically bring back to its baseline level by applying dasatinib, and this is also true for the late activation marker CD25. So after this, we tested a different strategy of administering the dasatinib. So first we tested applying dasatinib throughout 8 days. So basically from starting the perfusing the T cells, CAR T cells. And what we could see here is that we were actually preventing the cytokines from being released, from peaking on day 1, and then remain low over time over the period of 8 days. Here exemplified in TNF-alpha and interferon gamma secretion. And in interferon gamma, the values are actually below even compared to the control T cell level. But what happened to the infiltration is that actually we are preventing the CAR T cells to infiltrate the tumor and then not controlling their growth. So this is not the condition that we want for the patient, right? So then we tested to apply dasatinib from day 0 to 1. Here we could see that the cytokine release is being delayed. For example, here interferon gamma, although not TNF-alpha. And here we could again see that the CAR T cells were barely infiltrating the tumor. But when we apply the dasatinib from day 1 to day 8, so basically starting from the peak of the cytokine release, we could see that we were actually able to bring down the cytokine level faster compared to the untreated condition. And what is interesting here is that we were actually also able to let the CAR T cells infiltrate first into the tumor aggregate within the first 24 hours, so before the dasatinib was administered. and then prevent them from overgrowing. So when we quantify the T cell intensity again within the tumor region of interest, we could see that we have half of the density of the T cells comparing it with the non-dasatinib condition. However, when we again measure the GFP intensity of both conditions, we were able to get no significant difference compared to the no dasatinib condition, meaning that we were able to actually also reach the baseline anti-tumor activity of the CAR T cells. So this is just an example of how we could basically use the system to test different strategy, meaning different duration and starting point of the administration of this pharmacologic switch off method. By testing that, we were able to basically find the perfect or optimal strategy to manage cytokine release without really diminishing the CAR T cell efficacy. So, which could be potentially also applied on other safety measure strategies. But in reality, the tumor is heterogeneous, right? So the next thing that we tested is whether or not the model can capture patient-specific effects. So what we are interested in answering here is whether or not the relative ROR1 expression level actually impact the T cell infiltration and cytokine response. So the strategy here is that we screen the cancer organoids biobank from the Women's Hospital at the University of Tübingen. Here we collaborate with Andre Koch's lab, and then identified several lines who are overexpressing ROR1 and then several lines who are not expressing ROR1. And then pick two lines, basically this one, the line 10, which overexpress ROR1. This is coming from a triple negative breast cancer specimen. And then the ROR1 negative is coming from the hormone receptor positive specimen. And then what we did then is we generate organoids from the single cells from these specimens and then put them into the chip and then applied the same CAR T cell perfusion throughout the system. So what we saw then is that in the ROR1 low patient-derived organoids or PDOs condition is that the tumor organoid actually merged over time and then continued to grow, and then we could barely see any infiltration into the organoid. Whereas in the ROR1 high patient-derived organoid condition, we could again see high infiltration of the CAR T cells and the tumor organoid stopped growing. And when we look at here again, we could better see how the CAR T cells actually colocalized with the tumor organoids in the ROR1 high patient-derived organoid condition on day 8, whereas in the ROR1 low condition, most of the CAR T cells were actually excluded from the organoids, which is similar to the control T cell condition. So we saw infiltration into the bottom tumor chamber, but not infiltration into the tumor organoid itself. Again, when we quantify the mean intensity of the T cells within the tumor, in our positive control condition, we could actually saw the highest infiltration, which is the MDA MB231 cell line. The second highest is the ROR1 high patient-derived organoid condition treated with CAR T, and then it remained low in the other conditions, right? So this shows that the relative ROR1 expression actually correlates with the T cell infiltration. And in terms of the cytokine response, again, we could also see correlation because we saw highest response in the positive control condition in green. And then in the ROR1 high patient-derived organoid condition, we saw also higher levels compared to the ROR1 low condition, meaning that we could basically capture this differences that is generated from the patient materials. So not only the differences by changing the target cells, we could also check whether the same modification of CAR T cells on different donor affect the cytokine response. For example, here, the same ROR1 CAR T cell, same modification from sourced from two different donors targeting the same target cells. We could see from the first CAR T cell donor, like a 10-fold cytokine response. For example, IL-2, IL-6, and TNF-alpha, and also mostly above the donor 2 at the measurement time points indicated here. So the differences in cytokine response is not only caused by the target cells, but also the quality of the CAR T cells. So whether or not this will mean it will be risky for the patients to have because of the cytokine release syndrome, we don't know yet, and this is something that we would have to investigate next. So just to sum it up, because now we can recapitulate the cytokine release syndrome, the next thing to really study is to check the contribution of autologous monocytes or macrophages in altering this inflammatory cytokine response, because these are the cell types that are actually secreting most of the cytokines clinically. What we are also looking at right now is to assess vascular leakage of a distant vasculature, which caused actually edema, for example, and other complications clinically. And here we collaborate with Andries van der Meer group. And as I mentioned before, we are now also looking into ways how we can benchmark the results here with clinical outcome to really say whether or not this model has a higher predictive value compared with the current model systems. So with that, I hope I can give some examples of how we can use the system to develop a safer and more efficacious CAR T cell therapy. With that, I would like to thank you for your attention. Thank you, Peter Laskill and the whole lab for this project, especially Claudia Teufel, Madalena Cipriano, and Lisa Razarevsky. For the patients as well, for the samples, and our collaborators from the clinic, Michael Hudecek and Miriam Out from Würzburg, André Koch from the University of Tübingen for the organoids, and Dr. Valeria Orlova and Lisa van den Hul for the iPS endothelial cells, and also IMSAVAR and EU Consortium for funding the project. And happy to answer any questions you might have.
Thanks. Thanks for this inspiring talk. Now there is time for questions.
My question is a technical one in regards to exclusion versus exclusion of which organoids you measure your CAR T infiltration. How did you decide which ones to include in your data analysis and which ones to exclude?
Yeah.
Did you exclude them all, or was there a standardized method that we as a community can agree upon?
Yes, so for the data analysis, I'm excluding all the organoid size below 50 microns and analyzing all of the sizes above that, basically. Yeah.
Other questions? Okay, otherwise we can go on with the next speaker.